Today we're going to start the innate immune system. And if you've listened to the lecture, which I hope you have, I know it's a testing period, it's overwhelming, but please do listen to the lectures before we start these lecture questions, because especially this topic, there's a lot of complexity. I do follow the book, as I mentioned last week, but I try and put it in, keep coming back to a bigger view, a bigger context. So I think that should help you as you're listening to the lecture, because it's amazingly easy to get lost in the little details, and that forest for the trees phenomenon can get pretty overwhelming. So I did assign the first lecture question, which is citing differences between the innate part of the immune system and the adaptive immune system. So, and I don't remember in the lecture if I talked about this, but some textbooks call the innate the non-specific immune response, and the adaptive the specific immune response. And as we go through these, when you come back from your groups, we could talk about that and what does it mean by specific. And the other part is, and why they named it innate, is because all creatures, that probably even plants, but we're just learning about that, but all creatures have some sort of immune response, because of course they are used as food sources by disease organisms, and basically that's what you are when a disease organism gets inside you and starts to grow inside you. So so the innate response is called that because all organisms may not work the same exact way that we have, but at least all mammals have a very, well, not all mammals, all chordates, meaning all animals with backbones, and below, even insects have an innate response. Once you get to the chordates, the animals with a backbone, that's where you see the first evidence of an adaptive immune response that looks somewhat like ours. So when we come back from the first group, then we're going to talk about the differences between the innate and the adaptive, besides in their name. Innate meaning, older evolutionarily, expressed by most organisms in maybe not the exact same way, but similar ways, and adaptive meaning more I guess evolutionarily advanced. That's kind of a maybe not a PC way of putting it, but that the chordates and above, vertebrates and above, have an adaptive immune response. Okay, so so I still only have 23 people here, but I'm going to go ahead and break you up. Can you put in the chat, if you remember, I know a lot of water has gone under this bridge, if you worked on that first innate question, you were in group 8 last Thursday, where you're citing the differences between the innate and the adaptive immune response. If you were, go ahead and put it in chat, and I will put you into that group. So the rest of you, I'm going to set up into breakout rooms, and I did post the questions in the announcements this morning. Sorry, I hadn't realized I hadn't posted them. So if you're not sure what your question is, go into that one. And I'm going to start with, we're going to only have 1, 2, 3, 4, 5 groups today. So we're going to have group E is the one that is from last week. And then A will be questions 2 and 3, if I remember rightly. And I've broken this up according to topic more than how many questions there are. So if there were related questions, I broke them up that way. So I'm going to make 5 rooms, and I haven't gotten anything in chat yet. So please do put down if you were in that group E last time, and I'll reassign you that because you've done work on it. Otherwise, I will just worry about whoever's left will end up in group E, I guess. So the first one I'm going to set up is going to be called A, group A. I'm just going to rename them now. This one E. It looks like you guys is, whoops, I didn't save it. E, yes, and E, and C. Okay, so I haven't gotten anybody saying they were in group E, so I'm just going to start assigning people into that first lecture question. So let's see, I have 26 people, so about 4 or 5 people in a group. So we'll just, I'll just go alphabetically. All right. And then I'll sign the next one. Sign the next one, which is sign the next one. 6. And that one, I think these last couple are a bit longer. And sign this last one. 6. All right, I think everybody has been assigned a room. Let me give you 15 minutes in the room. Okay. 15 minutes and so everybody's back, I think, and that first question is dealing with something I kind of introduced at the beginning, the innate versus the adaptive response. So we're stepping back, and the textbook does this as well, and looks at how the 2 parts of your immune system, what are their strategies, where are they acting, that sort of thing. So do you guys have a spokesperson? Anyone want to speak up for group E?
Oh, I thought we were starting with the
with A?
Yeah, with the last question from last time. I'm lost.
Well, what I think we had not done last time was the first question of the innate.
Okay.
That is just citing 8 differences between. Is that what you discussed, Dina?
Yeah, that's what I've got.
Okay. All right. And that was the last question. I'm going to go ahead and share my screen so I can take notes as you go along.
Okay. Well, I did my favorite when it comes to compare and contrast, and I did a table.
Okay.
So I've got innate. For each one, I'll start with innate, and then I'll move to adaptive.
All right. I'll try and set it up as a table. So innate over here. And adaptive over here. Oops.
Yeah. So I've got that the innate is primarily a local response with a nearly immediate response.
I don't know what's going on with my pen. Local and fast. Okay.
The adaptive is systemic. Okay. And for novel viruses, the response may take 7 to 10 days. Then I've got that the innate is always present, active, and ready, with circulating WBCs, first neutrophils, and then free macrophages. Tissue-specific WBCs are there with the fixed, dendritic, and mast in most connective tissue and epithelia. And then the adaptive, when the antigen pathogen becomes systemic, B and T cells found in the lymphatic tissue respond. Innate infection is in or on the skin mucosal surfaces, and I've got lymphocytes in parentheses.
Can you just say that one again?
Yes, the infection is in or on the skin and the mucosal surfaces.
Okay, so most likely entry. Okay.
Yes, entry. Yeah, entry or barriers.
Skin, but I'm going to put that in parentheses, and mucosal.
Yeah.
Skin's a pretty good barrier, so. Okay, yeah. And what would you putting on the adaptive side?
I've got the infection is systemic and it's found in the blood, the lymph, the tissues, etc.
I'm going to say multiple tissues. All right, good.
The intracellular pathogens, my notes don't make sense to me here. I've got intracellular pathogens, NK cells, interferons, some types of interferons and cytokines. And then I've got cytotoxic cells kill infected abnormal body cells on the adaptive side. And then I've got on the innate no memory. Last exposure elicits the same response as the first response or first exposure. There's a lack of learning. I have in parentheses that the lack of learning is now being questioned and disproved. And then on the adaptive, we absolutely have a memory. The first exposure is much weaker, and then ever increasingly stronger response with each subsequent exposure. And this is the basis for vaccines. And I just emphasize that the novel infections on the innate side, they still have a nearly immediate response, nearly immediate response, whereas novel pathogens, like I said before, on the adaptive side, take 7 to 10 days to respond.
Okay, so that's similar to what you said.
Yeah. And then I've got innate recognizes not self usually as a category rather than a specific. So their response is going to be non-specific to, which can lead to damage to self. And then the adaptive side has specific memory cells for specific antigens for each specific pathogen. So that's what I came up with.
Okay, that sounds really good. And it's all over the place, I know. Categories of pathogens.
Yeah, I didn't quite get it organized in flow.
Okay, so no, some of it's me organizing it and when I ran to the table here. So let's see if from that we can come up with 8 things just to help people fill this out. So one of the things was speed of their action, at least to a novel. Well, even if it's not novel. So innate is faster. That's one of the things she said, right? And especially if it's a pathogen that your body's never seen before, then your adaptive is going to be very slow to respond. So we also talked about location. Innate is mostly local, primarily local, at the site of infection. I'll put of entry because I can fit that, versus the systemic nature. Typically, and maybe I'll try and say that, typically the antigen, if it's not the whole pathogen, at least its antigen has to be brought into the lymphatic tissues. If it's MALT, those could be kind of local, but it's not right where the entry point is. It has to get into the lymphatic, versus adaptive, systemic, or at least in the lymphatic tissues. And that's partly because as you've heard, hopefully, as you'll see more later when we do the adaptive, most of the white blood cells that are involved in the adaptive response are in the lymphatic tissues. And if they're not, they have to go there in order to find other white blood cells that are waiting around in the lymphatic tissues. So there are a certain number of them that will circulate. Mostly they'll be T cells that'll be circulating. And then, whereas the innate, as Dina said, some of them are already out in the tissues even before the infection. And then the rest, the majority of them are the white blood cells circulating in your bloodstream. So let's see what else we have down here, where they are, who's faster, where they act mostly. And then we do have tools for intracellular, but the intracellular different. Now, what we mean by intracellular pathogens aren't always bacteria, viruses, fungi. It can also include things like cancers and abnormal body cells. So when it's intracellular, you need to come up with a strategy to actually attack your own body cells. So I'll call those intracellular, but that will also include things like abnormally acting body cells like cancers. Each have strategies, and you're going to see they're overlapping, but use different. I'll put ways of recognizing that the cell is abnormal. Because as you'll see when we get there, or you may have seen already if you've listened to the lecture, once they do, they often use some of the same tools to eliminate this abnormal cell, but they recognize that it's abnormal in different ways. Memory, that was the other thing. How the first time they see the pathogen may be different than the second time. So for the innate, you have primary, and secondary is basically the same. Characters are called up. They're called up in similar strength. Responses for the innate are basically the same. So as Dina alluded, there's now growing evidence that monocytes, macrophages, do become more honed so that there is a secondary response. But because they're right there and fast already, it's not like you see a speed or a strength difference. It has more to do with they'll be a little bit more specific and respond more readily because they've seen it. But it's not dramatic like you see versus secondary and adaptive. You're seeing a much bigger response, and it's much faster. Increased amount. I'll put. Okay, that's amount, by the way, in my trying to fit into my boxes here. So let's see, what else did Dina come up with over here? how they recognize. So this is where the specific and non-specific. So there are a couple features, and she brought up some of them. First of all, with the innate, they use categories, meaning these are called pathogen recognition receptors. That's PRRs. And you'll see that the textbook and my lectures are riddled with these acronyms. So good idea to make a flashcard or something to make sure you have these. PRRs recognize PAMPs. Now, what are PAMPs? And I went through this in the lecture. This stands for pathogen-associated molecular patterns. So again, another acronym. But basically what it comes down to are all bacteria, or almost all bacteria, have some of the same cell wall components. Be careful of distinguishing what a cell wall is from a cell membrane. Sometimes people get a little sloppy in their thinking. The cell membrane is, of course, the plasma membrane, the lipid bilayer. And the cell wall is something outside of that. So like the cellulose of plants is outside their plasma membrane. And not all bacteria, but many, many of them have cell walls. And those cell walls can have components which are not just one type of bacteria has, but many types have. So just like, doesn't matter if we're talking about a human cell or a mouse cell or a cat cell or a bird cell, you're going to see some of the same proteins in their plasma membrane. Say their cell-cell junction proteins, something like that. They'd be very common. They're conserved evolutionarily. Well, the same thing happens in microbes, whether they're bacteria or fungi. They commonly have cell walls that have the same sort of molecular building blocks. Now, white blood cells have receptors for these building blocks. So if you've had micro, then you may know about peptidoglycan, common to both gram-negative, gram-positive types of cell walls, at least to a certain extent, or lipid A and the gram-negative, lots of different microbes. Fungi use chitin in their cell wall. Doesn't matter the type of fungi. As long as there's a cell wall on the outside, it has chitin in it. So not only do they use that, but each white blood cell has many types of these PRRs. So Dina alluded to this. So each white blood cell that uses these with PRRs have many of them. I'll just put wide variety of them. So that is going to contrast with the adaptive, where each white blood cell, each B or T cell, has one unique antigen. And I'm even being a little sloppy here because the book does go into haptens, which are portions of the whole antigenic molecule. I'm not going to worry about that level of specificity. Just let's say one particular molecule is what this B or T cell recognizes, and only that one. Now, each bacteria may have many, many antigens on their outside, which means that many B or T cells can respond. Versus the innate, where you're going to have each white blood cell, say it's a macrophage or a neutrophil, it can respond to many, many different types. So let's say, just to be pretty broad, let's say I'm a macrophage, I could recognize a bacteria, I could recognize a fungi, I could recognize a virus. So as my one type of cell, I'm very non-specific. I have PRRs all over me, and some of them are going to recognize viral capsules. Some of them are going to recognize, the cell wall of a fungi. Some of them are going to recognize the various membrane or cell proteins unique to bacteria. So that's quite a bit different. And I just elaborated a little bit. Let's see if there's more things down here. I think that's as far. And somewhat I've shoved a lot of things in here. So I think even though, so another aspects of them that, as she said, is very similar. There's similarities and differences, but there's a term you're going to come across, cytokines, and both of them are going to use cytokines. Cytokines are signaling molecules where one part of the immune system talks to either another immune cell or often the liver or another part of the body, which gets it to mount an immune response. So these are immune signaling molecules. Now, they both use them, but in particular, the adaptive is very good at using these to enhance the innate. So we're going to put that down. So what I mean by enhance is you'll get a stronger innate response if you've got your adaptive. Now, one ways it does it is through signaling molecules, so I'll put via cytokines. But it can also do it with antibodies. Again, more on that later. On the other hand, the innate is almost always required before you can get an adaptive response. So the adaptive also depends on the innate almost always. There are some rare exceptions in order for it to be triggered. Oops. And this is going to be through a cell called an antigen-presenting cell. Again, another acronym, antigen-presenting cells. Excuse me. All right, so that was a big topic. Hopefully, is there anyone else in the group who wants to contribute anything? Because I'm sure we could probably come up with more differences or similarities, but it gives us a good comprehensive kind of view. So I'll give you guys a second. If there's anyone who wants to just do it in chat too, let me open chat so I can see if there's anything in chat. I know you had the test today, so it's a little, maybe everyone's still a little bit stunned from that. Okay, so before we go on, I have things here listed of the adaptive versus the innate. Again, similar to what Dina brought up, and you'll say, "Oh, well, can you give me all this?" Well, you know what? It's there already. And it may not look exactly like this, but there is a folder in your Canvas shell under Files when you go into the materials for the second lecture exam. So that folder has one that's labeled innate immune system. Open that because you're going to need some key things. You don't have to do it right now. But there are some things in there like concept maps, which is kind of what I roughly done here. Concept maps, if you remember from 103A, are a way of kind of organizing a lot of diverse information onto one page. You can do it with a table. That's another way of doing it. Tables tend to categorize things. Concept maps do too. But I'm just doing it here as bullets off to the side. Again, in our stepping back, trying to see this whole picture. The other thing you're going to find in there is a table that I constructed from the tables in the textbook, because one of the things, there's a lot I like about the textbooks dealing with the immune system, but one of the things that also kind of breaks up is all these various cytokines and antimicrobial molecules that you're going to be learning about are all in these tables that are scattered through the two chapters on the immune system. And so I just kind of put them together into one big table. And it's going to include cytokines you need to know, and it's going to include antimicrobials you need to know. Now, these are by no means comprehensive of what would be all of the immune system's cytokines or all of the immune system's antimicrobials. And in fact, for some of them, the book goes into a little more specific details than I do. I lump some of these things together. So one of the things that we haven't covered that I put in this little concept map, obviously some of the things that we have, like the innate primarily acting locally, is the three ways in which the innate does act locally in, again, categories that the book has broken up in a similar fashion. One of the ways is it causes inflammation. So it uses some of its cytokines to cause inflammation. Now, I'm not sure why. It seems to me that the book ought to do the inflammation first, since that is what's going to allow white blood cells to arrive that aren't already there. But it doesn't. So I'm following along in their order, because what it starts with is a discussion of phagocytosis. So now some of that phagocytosis at a low level is probably happening if you're in tissues that have fixed macrophages. So last time when we talked about the white blood cells, we talked about macrophages arising from monocytes. Well, there are some of them, including a type of phagocyte called a dendritic cell, that live within the tissue, especially connective tissues and epithelial tissues already there. So you can think of them sort of like and I hate to use this analogy these years, but it's the best one, I guess, like the border patrol agents. They're at the border of your body, and they are looking to respond early, but there aren't many of them, so they can't make a very big response. Finally, there are antimicrobials, and some of those are released by phagocytes, some of them are in your body secretions, and some of them are released in your plasma and activated during an immune response. So we talked about the rest of these. They lack memory, or at least not to the degree of the memory you see in the adaptive. And they provide antigen-presenting cells so they can trigger the adaptive. So this went to sleep. I'm going to have to wake it up again. Looks like I might have to share it again because it did go to sleep. Yeah, and then here we are. They use PRRs that bind PAMPs, and cytokines will also trigger it. And now it's not moving very fast, to recognize who is an invader and who is self. So in that sense, they are very non-specific, and that almost pretty much all the members of your innate cellular members can use, are non-specific in how they recognize non-self. Now, in the case when they're having to kill an abnormal body cell, they use a different mechanism, which is done by the natural killer cells, like Dina mentioned. And then finally over here, I say the adaptive supplements it, meaning adds members to it, and then enhances it, can make them more active, give them some, energizing juice. So I'm doing all of this, spending all this time, because I'd really like you to have a sense of where we're going. And then we're going to come back and do this in more details. And why is this There we go. It's slow. Just they're talking to each other, but very slowly. Maybe I'll have to stop share and start sharing again. Let me just try that. Let's see if this will respond a little fast. There we go. Okay, so as she mentioned, the adaptive slow, it's generally the antigen has to become systemic, at least reach the lymphatic tissues before you're going to get an adaptive, because that's where most of the cells are going to reside, or at least wander in and out of. And they'll need to encounter other cells within the lymphatic tissues in order to generate this. They require an antigen-presenting cell almost always, and they use a specific antigen receptor so that each of the lymphocytes in the adaptive response, which are the B and T cells, they have only one antigen that they can bind. It's unique to them. That's the only thing they're interested in, which means you need a whole variety of B and T cells. So each B and T cell has their own unique antigen. That means if we're going to battle all the potential pathogens in the world, some of which have multiple antigens on them, we need a big library of B and T cells. Now, when they do this, there's two steps that we'll come back to next week: double recognition and co-stimulation. And we'll talk about that. That has to occur. The last thing about it, which we haven't talked about, but we will next week, is you divide it into two types of responses, and Dina alluded to this. If it is an intracellular pathogen, you need a certain type of T cell. So the cellular response here those are called cytotoxic T cells, which I probably didn't write, T cytotoxic. These are for the intracellular or cancer. The other part of the cellular response, we're actually going to deal with this one first when we get to the adaptive. So I'm also going to rearrange the chapter in the book, because I'm going to put it in the order in which it really happens. They start with the humoral response, not with the cellular response, but you need the cellular response before you can get the B cell humoral response. So we have cytokines released by these T helper cells. These are the ones that are going to help not only enhance your innate that I alluded to before, but they are also required to coordinate and to initiate the rest of the adaptive response, again, almost always. And then we have these interesting regulatory cells called T regulatory that are important in ending the immune response, and we make memory cells. When we look briefly at the humoral is kind of a B cells staying in the lymphatic tissues that you saw before last week, and Drew will be drawing this week on Thursday, where you have these germinal centers, and the B cells become plasma cells in those germinal centers and secrete antibodies. There are 5 main antibodies that can be secreted, although it's not clear why IgD is secreted, so we'll really only talk about the first 4 in any detail. And these are long-lasting proteins that end up in your body fluids, circulating and seeking out a particular antigen. And again, we'll talk about their effects. And then lastly, besides the plasma cells, you make memory cells that cause the secondary response that is faster and stronger. And again, we'll come back to some of these details. So useful either taking screenshots of this, or which is a bit messy, or looking at the concept maps I put in that folder to kind of always step back, see where you are in the immune system. So there's one last thing we could have put in this table that is unique for, well, not unique, but is an aspect of your innate. And that is, there's parts of the innate that are not actually white blood cells or cytokines or phagocytes. It's actually the rest of the organs that are on the borders of your body. And that is the barrier system. So this is sort of the features of other organ systems, mucosal membranes or your skin, that act as a physical barrier to prevent entry. And that's the topic that the group A is going to be discussing, structural features that block the entry, the barrier part. And then once a pathogen does get into some of those borderlands, what are the immune-like cells in the borderlands that can start your battle? It's gotten through the fencing, and now you're starting with the border patrol to try and prevent this pathogen's entry into the rest of the body. So who are those cells? So does Group A have a spokesperson? You go past their table here. I should remind you guys to get a spokesperson, but I can go look and see who was in Group A if you guys pick somebody.
I can answer.
Who is this? Who is it?
Augustine.
Oh, okay, Augustine. Why don't you go for it then?
So what I have is the acid mantle.
Okay.
It's found on the skin and the vaginal and stomach secretion that inhibits bacterial growth. And I also have enzymes found in the lysosome that are found in saliva, in respiratory mucus, and the lacrimal fluid of the eye. So that destroys bacteria. And there's also protein digesting enzymes in the stomach that also kill a lot of microorganisms. And next one, I have mucin that dissolves in water, and that forms like a thick mucus that lines the digestive and respiratory passageways. The mucus traps a lot of microorganisms, and they I guess they can transport them to like the stomach, where they get digested or destroyed by stomach acid. And the next one, I have defensins that are also found in mucous membranes and in the skin, which they secrete some broad spectrum antimicrobial peptides, and they increase the response of inflammation when the surface barriers are breached, and they also disrupt microbial membranes and control bacterial and fungal colonization. And the last one I have is chemicals in the skin, like lipids and sebum and dermacidin, and in eccrine sweat glands. They're also toxic to bacteria.
Did you call them ergocycline?
I put chemicals in the skin like lipids and sebum, or dermacidin, I think.
Dermacidin.
Dermacidin.
Dermacidal, like that? Dermacidal?
Yeah, yeah, that's good. And
Okay.
Those are found in the sweat glands, and those are toxic to bacteria.
Okay. All right. So that's good. Your focus largely here were on what's secreted, and that would help prevent things or attack things that are on the surface before it gets into the living tissues. And in some cases, you also talked about secretions that you transport things to, like the mucin, you're working with cilia to move, cilia, to move a kind of flypaper trap through the sticky mucin of you, well, you know what mucus is like in your, say, your respiratory tract or in your oral cavity, and you're trying to move all that to your stomach and let the stomach kill it with its acids. So some things are trapping. And one of the other things I put or out is the book talks about is flushing. And I think that is kind of, or flushes, if I can fit that, kind of what you're getting at number. I don't know what the germicidal things are if they're not defensins. Maybe they were just saying, oh, there's a category, because sebum would certainly have lysozyme and defensins in them. And those are the only ones I expect you to know, fensins. But in addition, the sebum, et cetera, does flushing. So one of the themes of any of the borderland of your body is your skin is constantly releasing sweat. So it's actually even the water, as you think about it, just forcing things outward. Same with your urogenital tract. If you're always secreting mucus and that's flowing downward, which you know this because you got to change your underwear. And then in addition, when you urinate or defecate, you're pushing things always outward. So the bigger problem arises, and maybe that's why it shouldn't surprise you when you get sick, it often is through your digestive or respiratory tract, where you have to take things in and you have to exchange through a little thin membrane because you're trying to get something out of it, whether it's gases or food, substances. You need a thin, not a thick layer of cells. And therefore, often those areas are most vulnerable. So once again, you're going to use the mucins and the enzymes that he mentioned, but you can't flush them outward. So instead, you send them to your stomach if you can. So what about some of these things he mentioned? Lysozyme, as you might have gotten from the lecture, is particular against a certain type of bacteria called gram-positive bacteria. They have, sometimes this doesn't want to write. They have a substance called peptidoglycan. Now, none of our cells ever make peptidoglycan, and this is in their cell wall. There is peptidoglycan in the other kinds of bacteria, cell wall, gram-negative, and then there are bacteria without any cell wall that wouldn't be affected by this. So lots and lots of bacteria have this, lots of different types of bacteria that are gram-positive have on the outside peptidoglycan. The others kind of hide their peptidoglycan a bit. But this enzyme specifically attacks that substance, and therefore it can get rid of a lot of, it's an enzyme that can get rid of a lot of bacteria without affecting our cells because we don't make peptidoglycan, so it doesn't have its particular, won't bind to its active site. Defensins, notice the S. This is a big category. So I know as he covered this, he gave a lot of different things it does. Antimicrobial peptides, decreases microbe growth, attacks the microbes, because there are many types of them. But in general, this is going to try and suppress microbes numbers on the outside of your body because it's released into the body secretions. So this is in the body secretions. Now later we'll learn it's also released by some white blood cells once you start an innate response. To release it. So good. So that sounds really, really good. The other things I would point out that if you're in epithelial cells, you're going to have lots of tight junctions. So we also want to make a structural wall. So Augustine really did a fantastic job of going through like what is secreted and what in those secretions would help eliminate things. But we also just really want to build a physical wall. So epithelial's tight junctions, it doesn't matter where you are, whether you're a genital, your skin, you're going to have lots of tight junctions to try and prevent, to decrease the paracellular route when you can. Now, once again, when you're in your digestive tract, you're going to definitely decrease it in your stomach. You don't want the stomach acids, but and pretty much in your intestines, that's going to be true as well, where you have very little or no paracellular between the cells. So anything that goes through has to go through by crossing the plasma membrane of those epithelial cells. So along those lines, if it's the skin is actually a really good barrier. And if you go back and review the skin, you know you have many dead cells. So you actually kind of coat yourself with a whole bunch of dead bodies that are dry with no or few nutrients for things to grow on. I mean, despite that, microbes are really good. So you have quite a variety of microbes that will grow on your dead skin, but you're trying to keep those microbes as far away from the living tissue as possible on your skin, and they'd have to make their way through wade their way through all these dead cells to get to anything living. And brings up one other feature we talked about in 103A, and that is that epithelia is avascular because the blood supplies nutrients, but also acts as a highway system. So if a microbe gets into the blood or the lymph, it then has access to other organs. So by having epithelia be avascular, then you are preventing it from entry as much as possible. Okay, so I think that's good for now. We will see later that amongst the body secretions, you're going to also have some antibody secretions called IgA, but we'll come back to that later. So that's a way your adaptive, again, is enhancing this wall that you're building. So I'll put it on here, but we won't talk about it too much. IgA in the secretions. I also neglected things like urea on your skin. Remember, you have some microbes. When you take microbiology, hopefully you get to actually take it in person, you'll see you use plates that have urea in it because many bacteria don't like urea and will not grow. Of course, there's some bacteria that will, but you add other things, high salt levels. So many bacteria, again, don't like salts, and many fungi won't either. It's an osmotic challenge. So for the skin, you've got other things like urea, high salt levels that make it osmotically challenging. All right, so the second part of this was, okay, now it gets through this barrier. Who are two cells that it might encounter once it gets in there? And is that you again, Augustine, or is there someone else who wants to step up to the plate? You got a shy group there.
I can answer.
Okay.
Yeah, I have neutrophils, macrophages, and monocytes that are phagocytic, and they confront pathogens and consume them.
Well, we actually want not the circulating cells in the blood. We want the cells that are already in the epithelia tissue. Did anyone else, or?
I put natural killer cells.
Yeah, those could be in there. So I'll put those down. Sorry. And this is something that Dina alluded to. There are fixed macrophages. So yes, they at one point were a monocyte, like you brought up, but they were not triggered by an infection to come in there. Most of these fixed macrophages are actually dendritic cells. It's not clear yet if all of them are, but most of them are dendritic cells. So they come from that same lineage, which is the monocytic lineage coming from the myeloid stem cell, the parental stem cell. But now we believe they break off into not just making the monocytes that become the macrophages, but they become a specific kind of macrophage called a dendritic cell. And it's called that because they have really long extensions like dendrites on a neuron. It's just how they look. It's not what they do. Now, these are both phagocytes. Now, whether all of the fixed macrophages are dendritic cells is a little muddy. I even tried to research this last week, and I think it's just not well understood yet how many of them are going to become just a pure macrophage. And how are they distinguished? Well, these guys are mostly for coordinating and killing, coordinating the innate and killing. And these guys are mostly going to become antigen-presenting cells. Now, they do kill the pathogen in order to do that, but it's their main focus is to go and try and get the adaptive initiated, as opposed to staying locally and doing the job locally of killing and then sending out cytokines to coordinate other aspects. So NK cells we are going to come back to, but remember from what we said before, we'll come back to these later. These are for intracellular and cancers, and they can be largely in connective tissues. There is a third cell, and the book is pretty wishy-washy about whether this is a white blood cell. Again, I'm not sure it's totally known, but my guess is that it is. And I've certainly seen it in online, researching online as a type of basophil, but we're unclear whether basophil lineage makes these cells, but they certainly act the same. And these cells we talked about very briefly in 103A. They're called mast cells. So mast cells are the two that we're going to be kind of focused on, and the book does, as well as the ones residing and battling any kind of extracellular, or for that matter, viruses too, they can battle, would be these macrophages and these mast cells being triggered. And then later we'll discuss what happens after the virus invades a body cell. Then you have to have a natural killer cell. But sort of as the very initial response, when we have just pure virus or bacteria or fungi that's gotten through your barrier, and now it's in the tissues, the first ones to act are going to be these fixed macrophages and the mast cells. Okay, so then it says state how each of these acts. So again, Augustine, what is a job of a macrophage? So maybe I'll go down here. What is it acts by? I kind of told you already, by killing, but exactly how does it kill? Do you have any inkling?
Is it phagocytosis?
Yeah, mostly phagocytosis. These stay for the big eaters. They also release defensins, lysozyme, if they're a macrophage. If they're a dendritic cell, I'm not clear if they release those things, but they're definitely phagocytes. So we'll put the phagocytosis on that side. What about mast cells? What do they do? Does anyone know? They release something, and basophils release the same thing. And we went through what basophils do.
They release histamine to increase inflammation and start
Right.
vasodilation.
So these guys are not attacking the pathogen. What they're doing is they are opening up, they're calling in and then opening up the spaces between the endothelial cells and the capillary beds there so that the other white blood cells that are circulating will be able to come out. But also, besides the white blood cell, plasma will leak out. That's what inflammation is about. That's why you feel it's hot, because you get vasodilation, more blood runs to that area. It's swollen because fluid leaks out through the paracellular routes that have opened up. So you've made that blood vessel leaky. So even without changing blood pressure, HP there, it's easier for fluid to go be driven out. But you vasodilated, so HP went up. More stuff comes out. It's still not going to allow really large proteins out like albumin, and it's not going to allow cells that aren't white blood cells from leaving, but it is going to allow them to open up, and the white blood cells are going to change their nature. They're going to bind to the endothelia and they're going to crawl out. So the next group, I think we're good there. We can come back if we need to. But the next group is now talking, let me change colors. So is talking about these PRRs and PAMPs that I discussed. And then we're going to look at the cells that arrive from the blood. So these are going to be called free macrophages, meaning that they are circulating as monocytes and then they're called out of the blood to become a macrophage, to mature and become an active macrophage. And we're going to be looking at neutrophils and starting, and this is going to start to, if you've looked at them, be a little familiar of just beginning the first essay. It won't be enough to complete the entire first essay. You may have to spread this over time, because we won't get to dendritic cells until much later, and that's in there as well. So it does ask you to compare the free and the fixed macrophages, but most of those fixed ones are probably dendritic cells. Okay, so let's talk about the who's the spokesperson for question 4, which is on the PRR and PAMPs.
That would be me, Jeanette.
Okay, Jeanette.
So you basically already said what we discussed for 4, that PAMPs are pathogen-associated molecular patterns. And when you're looking at a lot of bacterial cell walls, you'll notice there's peptidoglycan, there's lipoteichoic acid, and that's very unique to a lot of these bacterial cells. So PRRs, the receptors on our white blood cells, will recognize a lot of those patterns and initiate the immune response.
Okay, so we gave a couple examples. You said peptidoglycan, and did you mention something else besides?
Lipoteichoic acid.
Oh, yes, that's that's another one. Okay, so these guys are fungi, but the others are all bacteria. And if it was a virus, you would have the capsomere proteins. Again, more on this when you, oops, spelled it wrong. Capsomere proteins. So these are kind of all what you find on the outside. These are PAMPs. And then, as she mentioned, you have the PRRs. But each PRR, you're going to have a whole variety of these, is specific to, one of the things on the list over here. So this white blood cell has is just studded with lots of different receptors all over it. And that means that that particular white blood cell could respond to a whole variety of different pathogens. So, who has PRRs? Did you discuss that?
No, we just generally said the white blood cell. It would be, sorry. It would be part of the next question where neutrophils and macrophages both have PRRs, but the difference was that neutrophils use the PRRs, but can also be independently activated by the cytokines.
Oh, great. That's great. Okay. So we'll come back to them in a second. Macrophages. So, and we're going to include in that the dendritic cells. So this is both fixed and free, since the book kind of mushes the fixed with the macrophages. Dendritic cells is all kind of a blurry thing. But remember, free are the ones that are called in from the blood, and fixed were already there. Now there is one other cell, and it's already there as well. What cell is already there that I mentioned just a minute ago?
Was it the dendritic cell?
No, that's not on here already. It starts with an M.
Mast.
Monocytes?
Mast cells. Mast cells have PRRs as well. Because after all, if we want to start inflammation, we need to have them be able to recognize there's something wrong here. So these all have, and someone just mentioned monocytes. Yes, they do, because monocytes become this. I don't know actually if when they're circulating, they have their PRRs out or not, because they do have to take about 12 hours for them to go from being an immature monocyte circulating in the blood to becoming the macrophage that's the active form, because the monocyte is immature. So I'm not sure if they're actually got their PRRs out, because they're not going to respond when they're circulating in the blood. They have to crawl out of the tissue, and then they can respond. So there is another cell, eosinophils, probably also have PRRs. Now, again, the book does not mention these guys much. So I'm going to put that in parentheses, because we're probably not going to really talk about it much as that. Now, the other thing that Jeanette brought up, which is really critical, is some of these PRRs binding their PAMPs is what initiates to trigger, right? But some of these have other ways of being triggered. So again, very non-specific ways. This is the non-specific part of your immune system. So, so it's not surprising that you want to have more flexibility. This may be the right word. So in this case, you can have cytokines. Some cytokines will trigger the neutrophils. In particular, things like prostaglandins released from damaged cells. So you don't even have to wait till you get an activated immune cell releasing the cytokine, the damaged cell membranes. So you cut your finger, some of those damaged cell membranes are going to release echinocyannoids, which are little fatty acids that fall under the category of prostaglandins. And they can, if a neutrophil comes into the area, that alone can get that neutrophil to start to release its granules. Same with mast cells. They can be triggered by the prostaglandins, and they can also be exaggerated by antibodies that will come come, I don't know where to write that. Whoops, I just scrolled right past there, but plus antibodies, I'll put here. IgE. So these are not the only thing that can, only actions cytokines have on these cells, but we're focusing here is on how do we trigger them? How do we go from the neutrophil that's maybe invaded the space, it's crawled out of the bloodstream, but now how does it know when to start releasing its granules and acting as a phagocyte? And in order to be a phagocyte, you always have to use your PRR. So the PRR binding to the PAMP allows for phagocytosis, permits phagocytosis. If you don't have that, all the cytokines are going to do is let you release whatever granules you have. So if you're a macrophage, you might have some defensins or lysozymes inside of you, and cytokines could get you to release more of those things. But in order to actually phagocytize, you have to bind. You can't just, I know in introductory biology, it looks like they just kind of gulp it, like you would open your mouth or a whale would open its mouth and eat the fish. That's not what happens. It has to actually physically bind a protein on the membrane of the phagocytic cell to something on the exterior of the pathogen or the microbe, and then it can bring that into it to permit phagocytosis. Now, why I bring this up, we'll come to in a second, I think. It's in the next question. Well, we haven't done all the differences between the neutrophils and the macrophages. So we'll come back to this idea. Let's talk about neutrophils versus macrophages, because this is an important thing for the, oops, go away. Oops, sorry, there we go. For this essay, because you are going to be working on this part. So let's see how much time we got too. We don't have much time. So let's just at least get started on this. Sorry, I went a little bit slow with all this stepping back. How about, is there anybody in group B that wants to speak up on more differences? I heard some from Jeanette.
I can just continue it for the group. I guess neutrophils are first to arrive, and they're most numerous. Although they're most numerous, they actually cannot discriminate between the self and non-self. So they can destroy. They also destroy your own cells. So they release hydrolytic enzymes that damage the pathogen, but also our own cells.
Okay. And then early on, you said they can be triggered by cytokines.
Mm-hmm.
Okay. They're early to arrive, probably in part because they're most numerous of all of them, right? Like 50 to 70% of white blood cells. But they are also mature when they arrive, whereas unless you're a fixed macrophage, which you could be, I'll try and do this a different color, fixed or low numbers already present, and then they're probably a dendritic cell, but again, we don't know if all of them are. Otherwise, slow to arrive. from the blood. Partly because they're so low in numbers, but also because and must mature, which you don't have to do if you are a neutrophil. Now, when they release things from their granules, just like she said, there's some things that they both release that are quite similar, and we'll go back to that, but they do not release hydrolytic enzymes. So that means that they don't tend to damage your own body cells, whereas neutrophils do. So neutrophils are, there's a little cute video, I've got to put that up on it, it's a link, where they call these the duck hunters with an Uzi, with a semi-automatic rifle. The duck dies, but so do a lot of other things. Whereas the macrophages that are in there and the dendritic cells, they are more selective. What they release, and both of them release, so something that both share, are going to be some of the things that Augustine brought up. So both release maybe I shouldn't have had a line here. Both release the defensins that are more specific against pathogens, don't act on our own cells. They both release lysozymes. Same idea. They act on gram-positive bacteria, not on our own body cells. And they both release pro-inflammatory substances and go away. Inflammatory substances like, and here's our first interleukins. Leukotrienes. I'm saying one thing and spelling something else. So whenever you see an S at the end, that means there's more than one type of them. We're not worrying about it. We're going to lump them all together. See the word leuko here, like white blood cell? These are causing inflammation. Now, please, since we're out of time now, this sort of thing, these sorts of things, they are in a table. You need to know this table. So on your essays, the top of the page with the essays on it also says you need to know a table that's in that folder on the immune system. It's called innate molecules. And there are two versions of it. One is empty. I put that in there so that you can write in your own words into the boxes. And then the other is the key, which of course I wrote things or I typed things in there. But I strongly recommend that you try and write to help learn. They're both there. So one is a tool that you can write things yourself. You could, this is that fusion of tables that I got out of the book. So you can also find information in the book that's on all these substances. Now, what do you need to know? The source of them, who releases them. So we now know that both neutrophils and macrophages release defensins, lysozyme, but so do tear ducts. So do sweat glands. So does mucous membranes release those things. On the other hand, leukotrienes are released only by white blood cells, and those are going to include both neutrophils and macrophages. So we're just starting that first essay. You can work on it on your own, and I probably won't post input on it for another week, maybe. But we are just starting that first essay. But you also need to know that table, and that table might help you supplement that essay. Okay, so it looks like we didn't finish all of this, but we didn't finish the Define the opsonization, phagosome, lysosome, and respiratory burst. Maybe one of your group can do that when we come back. I won't even assign it. We'll just start right there, and I'll pay attention to who's in that group. And then the other group C, if you guys can kind of stay together, it would be probably move things along. Otherwise, I will just go ahead and assign you into that group. Okay, and did we have a D? I can't remember now. Did we get that far?
Yeah, we did have a D
Oh, we
group.
did, yeah. So if group D, we'll start with those two groups and that last question when we come back on Thursday. All right, so I will, if you wish, I'll be here. So I'm not going anywhere. If you come to the next lab session, you're welcome to do that. And let me see if there's anything in, oh, people are putting the answers to things in. That's nice into chat. Okay, so I will see you otherwise on Thursday, and I'll have posted these groups now that we see where we are at on Thursday. Okay? All right, have a good rest for the next couple days. No tests. All right. Stop share, and I will see you in a few days.